The Order of Life, Navigating Biological Classification and Systematics. The living world is a tapestry of bewildering complexity and astonishing diversity. From the microscopic bacteria that inhabit every corner of our planet to the colossal blue whales that navigate its oceans, life presents an almost infinite array of forms, functions, and behaviors. Faced with such an overwhelming panorama, the human mind instinctively seeks order, patterns, and categories. This fundamental impulse to organize is not merely an aesthetic preference. It is a critical tool for understanding, predicting, and communicating about the natural world. This is the domain of biological classification and systematics. At its core, biological classification is the process of arranging organisms into groups or taxa, singular, taxon, based on their shared characteristics. It's like sorting a massive library, where books are grouped by genre, author, or subject. But in biology, these groupings are not arbitrary. They reflect real, underlying relationships, revealing the intricate web of kinship forged over billions of years of evolution. Systematics, a broader and more encompassing field, is the study of the diversification of living forms, both past and present, and the relationships among living things through time. It includes classification but goes beyond it, actively seeking to understand the evolutionary history, phylogeny, that explains why organisms are related in the ways they are. Imagine trying to discuss a specific species of orchid without a standardized name, or trying to understand the spread of a disease without knowing the relationships between different pathogens. Without classification and systematics, biology would be a chaotic collection of observations, lacking the coherent framework necessary for scientific inquiry. These disciplines provide the vocabulary and the grammar for understanding life itself. Our journey into biological classification begins not in a laboratory with DNA sequencers, but in the distant past, driven by practical needs. Early humans needed to distinguish edible plants from poisonous ones, dangerous animals from docile ones. This required a rudimentary, often local, classification system. As human societies grew, so did the need for more formalized systems. Aristotle, the ancient Greek philosopher, made one of the earliest recorded attempts at scientific classification, grouping animals based on characteristics like red-blooded versus non-red-blooded, and dividing plants into trees, shrubs, and herbs. While rudimentary by modern standards, it laid a foundation, the idea that organisms could be grouped based on observable traits. However, the real revolution in classification came in the 18th century with the Swedish botanist Carl Linnaeus. Before Linnaeus, the naming of organisms was chaotic. A single species might have multiple long, descriptive names that varied from region to region. This made consistent communication impossible. Linnaeus, in his seminal work Systema Naturae, introduced a standardized, hierarchical system that we still use today, albeit with significant modifications. Linnaeus's genius lay in two key innovations. First, he popularized binomial nomenclature, the system of giving each species a unique two-part scientific name. The first part is the genus, always capitalized, and the second part is the specific epithet, always lowercase. Both are italicized. For example, humans are Homo sapiens. The lion is Panthera leo. This simple yet powerful system provided a universal language for naming species transcending geographical and linguistic barriers. It's concise, unique, and universally recognized. Second, Linnaeus established a hierarchical classification system. He grouped species into increasingly inclusive categories, species into genera, genera into orders, orders into classes, and classes into kingdoms. His original hierarchy was kingdom, class, order, genus, species. This layered approach allowed for a systematic way to organize vast numbers of organisms. It provided a framework for understanding not just individual species, but also their relationships to broader groups. Crucially, Linnaeus's system was based primarily on shared morphological characteristics, what organisms looked like. He recognized that organisms with similar physical features often shared common ancestry, though he himself did not formulate his system with evolution in mind. He believed in the fixity of species, seeing his classification as revealing the divine order of creation. It was only later, with the advent of Darwinian evolution, that Linnaeus's hierarchical system found its deeper, more profound meaning. The dawn of evolutionary systematics. Charles Darwin's theory of evolution by natural selection, articulated in On the Origin of Species, 1859, fundamentally transformed biological classification. Darwin showed that the similarities between organisms were not just superficial resemblances or reflections of a divine blueprint, but rather evidence of shared ancestry. His famous phrase, descent with modification, provided the theoretical underpinning for systematics. The hierarchy that Linnaeus had observed was now understood as a reflection of the tree of life, with branches representing lineages diverging from common ancestors over geological time. With evolution as the guiding principle, the goal of classification shifted from merely cataloging diversity to reconstructing the evolutionary history of life. This new approach is called phylogenetic systematics, or sometimes cladistics, from the Greek klodos, meaning branch. The aim of phylogenetic systematics is to group organisms based on their evolutionary relationships, meaning that all groups, taxa, should be monophyletic. A monophyletic group, or clade, includes a common ancestor and all of its descendants. This is the ideal for modern classification. To illustrate, birds, for instance, are the direct descendants of dinosaurs. To make the group reptiles monophyletic, it must include birds, or else it is a paraphyletic group, a common ancestor in some but not all of its descendants, like reptiles without birds. A polyphyletic group, by contrast, consists of descendants from more than one common ancestor, often grouped together due to superficial similarities, e.g., grouping flying animals like birds, bats, and insects would be polyphyletic. Modern systematics strives to eliminate paraphyletic and polyphyletic groups from formal classification, replacing them with monophyletic clades. To reconstruct these evolutionary relationships, systematists look for homologous characters. Homologous characters are similarities between organisms that are due to shared ancestry. For example, the four-limb bones of a human, a bat, a whale, and a cat are homologous. They have different functions but share a common underlying structure because they were inherited from a common vertebrate ancestor. In contrast, analogous characters are similarities that arise due to convergent evolution, where different lineages evolve similar features independently because they face similar environmental pressures. E.g., the wings of a bird and the wings of an insect are analogous. They both facilitate flight, but they evolved independently. Distinguishing between homologous and analogous characters is crucial for building accurate phylogenies. The process of building a phylogeny typically involves several steps. Character selection. Identifying a set of characters, morphological, anatomical, developmental, behavioral, molecular, that can be compared across different taxa. Character states. Defining the different forms each character can take, e.g. for presence of feathers, the states are present or absent, outgroup comparison. Including an outgroup, a taxon that is known to be less closely related to the group of interest than the members of the group are to each other. This helps to determine the ancestral, primitive, versus derived, evolved states of characters. Cladogram construction. using computer algorithms to analyze the distribution of shared-derived characters, synapomorphies, among taxa. This generates a cladogram, a branching diagram that visually represents the hypothesized evolutionary relationships. Each branch point, node, represents a common ancestor. The principle of parsimony, Occam's razor, is often used in cladistics. The simplest explanation that accounts for the observed data is usually preferred. In phylogenetic terms, this means preferring the cladogram that requires the fewest evolutionary changes, character state transitions, to explain the distribution of characters among taxa. More recently, statistical methods, particularly maximum likelihood and Bayesian inference, have become popular. These methods estimate the probability of a particular tree given the observed data in a model of evolution, often yielding more robust and statistically defensible phylogenies. The modern tools of systematics. Initially, systematists relied heavily on macroscopic morphological features, the shape of bones, the arrangement of leaves, the number of petals. While these remain important, the advent of molecular biology has revolutionized the field. Molecular systematics uses genetic data, primarily DNA and RNA sequences, to infer evolutionary relationships. The logic is simple. The more similar the DNA sequences between two species, the more recently they shared a common ancestor. DNA sequences change over time due to mutations. By comparing the number and type of differences in specific gene sequences, or even whole genomes between species, we can estimate their evolutionary distance and reconstruct their branching patterns. Molecular data has several advantages. Abundance. Every organism has DNA, providing a vast amount of data for comparison. Objectivity. DNA sequences are less prone to subjective interpretation than morphological characters. Quantitative analysis. Genetic distances can be precisely quantified. Resolving cryptic species. Molecular data can reveal cryptic species. Species that look morphologically identical but are genetically distinct and reproductively isolated. Deep time resolution. Some genes evolve very slowly, making them useful for resolving relationships among very distantly related groups, e.g., across kingdoms. Other genes evolve rapidly, suitable for distinguishing closely related species. The most common molecular markers used in phylogenetic studies include ribosomal RNA, rRNA genes, particularly 16 srRNA in prokaryotes and 18 srRNA in eukaryotes. These genes are universally present and evolve slowly, making them excellent for resolving deep evolutionary relationships. Mitochondrial DNA, mtDNA. In animals, mtDNA is maternally inherited, evolves relatively rapidly, and is very useful for resolving relationships among closely related species and populations. Chloroplast DNA, cpDNA. In plants, cpDNA has a slower evolutionary rate than mtDNA and is useful for plant phylogenetics. Nuclear genes. A vast array of nuclear genes are used, selected based on their evolutionary rate and presence across the taxa of interest. With advances in genomics, whole genome comparisons are becoming increasingly common. The power of molecular systematics has been immense. It has confirmed many relationships inferred from morphology, resolved long-standing controversies, and uncovered entirely new evolutionary relationships that morphology alone could not reveal. For example, molecular data showed that fungi are more closely related to animals than to plants, a conclusion that was initially surprising given their superficial resemblance to plants. It also revolutionized our understanding of microbial diversity, revealing vast lineages of bacteria and archaea previously unknown or misclassified. The hierarchical ranks and the kingdoms of life. Linnaeus's hierarchical ranks, kingdom, class, order, genus, species have been expanded and refined over time to accommodate the increasing complexity of life's tree. Additional ranks like phylum between kingdom and class, family between order and genus, and various prefixes like super and sub, e.g., superorder, subgenus, are now routinely used. The full hierarchy typically looks like this, from most inclusive to least inclusive. Domain, kingdom. Phylum or division for plants and fungi, class. Order, family. Genus, species. Each of these ranks represents a clade, or at least aims to in modern phylogenetic classification. The species is generally considered the fundamental unit of classification, though its definition can be surprisingly complex. The biological species concept, popularized by Ernst Mayr, defines a species as a group of individuals that are actually or potentially interbreeding natural populations that are reproductively isolated from other such groups. However, this definition doesn't work for asexual organisms, fossils, or situations where hybridization occurs. Other species concepts exist, such as the morphological species concept, based on shared physical traits, and the phylogenetic species concept, based on smallest diagnosable monophyletic groups. The choice of species concept often depends on the type of organism and the specific research question. Perhaps the most significant change to Linnaeus's original hierarchy has been in the highest ranks, the kingdoms. Linnaeus initially recognized two kingdoms, Animalia and Plantae. As microscopy revealed the world of microorganisms, this system proved inadequate. Two kingdoms, Linnaeus, Plantae, and Animalia. Three kingdoms, Haeckel, 1866, Protista for single-celled organisms, Plantae, Animalia. Four kingdoms, Copeland, 1956, Monera, for bacteria and archaea, as prokaryotes lack a nucleus, Protista, Plantae, Animalia. Five kingdoms, Whitaker, 1969, Monera, Protista, fungi, recognized as distinct from plants due to their heterotrophic nature and cell wall composition, Plantae, Animalia. This five-kingdom system became widely adopted in textbooks and remained influential for decades. However, molecular data, particularly comparisons of ribosomal RNA sequences by Carl Woese in the late 1970s, revealed a profound split within the Monera. He discovered that prokaryotes were not a single, unified group, but rather comprised two fundamentally distinct lineages, bacteria and archaea. These two groups, while both lacking a nucleus, differed dramatically in their genetic makeup, cell wall composition, and metabolic pathways. This groundbreaking discovery led to the proposal of a new highest rank, the domain. The three-domain system, Woese, 1990, is now the most widely accepted classification at the highest level. Domain bacteria comprises the vast majority of prokaryotes, including familiar forms like Escherichia coli and cyanobacteria. Domain archaea includes prokaryotes often found in extreme environments, extremophiles, but also abundant in less extreme habitats. They share some genetic similarities with eukaryotes that bacteria do not. Domain eukarya includes all organisms whose cells have a nucleus and other membrane-bound organelles. This domain encompasses the traditional kingdoms of Protista, a polyphyletic group now often considered several distinct kingdoms or supergroups, fungi, Plantae, and Animalia. This three-domain system is a powerful illustration of how systematics evolves with new scientific understanding. It reflects a fundamental evolutionary divergence early in life's history, showing that the split between bacteria, archaea, and eukarya is more ancient and significant than the splits between the traditional eukaryotic kingdoms. Beyond the ranks, the tree of life. While hierarchical ranks are useful for organizing and retrieving information, they can sometimes obscure the true branching nature of evolution. The ultimate goal of systematics is not just to assign names or slot organisms into categories, but to reconstruct the entire tree of life. This tree is a grand hypothesis of life's evolutionary history, depicting the divergence of all species from common ancestors. The tree of life is constantly being refined as new data emerges. It's not a static entity, but a dynamic, growing scientific model. Molecular data, especially genomic data, has been instrumental in illuminating previously obscure branches of this tree. For example, it has shown that eukaryotic life itself is incredibly diverse, comprising many supergroups that are as distinct from each other as animals are from plants. The traditional kingdom, Protista, is now recognized as an artificial polyphyletic assemblage that needs to be broken down into multiple distinct lineages. Ongoing large-scale genomic projects aim to sequence the genomes of thousands, even millions of species, providing an unprecedented amount of data for phylogenetic reconstruction. The challenge then shifts from data acquisition to developing sophisticated computational tools to analyze these massive datasets and construct ever more accurate and comprehensive trees. The practical importance of systematics. Beyond satisfying intellectual curiosity, biological classification and systematics have immense practical implications across many fields. Conservation biology. To protect biodiversity, we first need to know what species exist, where they live, and how they are related. Systematics provides the framework for identifying endangered species, understanding patterns of biodiversity, and informing conservation strategies. For instance, knowing the phylogenetic relationships between different populations of a species can help prioritize which populations are most genetically distinct and therefore most critical for long-term survival, medicine, and public health. Understanding the classification and phylogeny of pathogens, bacteria, viruses, fungi, parasites, is crucial for tracking disease outbreaks, identifying new pathogens, developing vaccines and treatments, and understanding drug resistance. For example, phylogenetic analysis of viral genomes can trace the origin and spread of pandemics, like influenza or SARS-CoV-2, agriculture, and food security. Classification helps identify crop-wild relatives that may possess genes for disease resistance or drought tolerance, which can be bred into cultivated varieties. Systematics also aids in identifying pest species and their natural enemies, informing strategies for biological control. Drug discovery. Many pharmaceuticals are derived from natural products. Systematics can help prioritize which organisms to screen for novel compounds by targeting phylogenetically diverse groups or relatives of known medicinally important species. Bioprospecting. Discovering new genes, enzymes, or metabolic pathways for industrial applications, e.g., biofuels, bioremediation, often begins with identifying novel organisms through systematic surveys. Ecology. Ecological studies rely on accurate species identification to understand community structure, food webs, and ecosystem functioning. Evolutionary biology. Systematics is the foundation of evolutionary biology, providing the tree upon which evolutionary events, e.g., the evolution of flight, the loss of limbs, the development of complex social behaviors, can be mapped and studied. It allows us to test hypotheses about adaptation, speciation, and macroevolutionary patterns. Biogeography. Understanding the evolutionary relationships of species helps explain their geographical distribution patterns, providing insights into geological history, climate change, and dispersal events. Challenges and controversies in systematics. Despite its advances, systematics is not without its challenges and ongoing debates. The species concept, as noted, defining a species remains complex and context-dependent. The biological species concept, while useful for sexually reproducing animals, often falls short for asexual organisms, plants, which frequently hybridize, and bacteria. The ongoing debate about how to delineate species boundaries continues to shape taxonomic practice. Horizontal gene transfer, HGT. In prokaryotes, bacteria, and archaea, genetic material can be transferred directly between unrelated organisms, not just vertically from parent to offspring. This phenomenon, known as horizontal gene transfer, HGT, can make reconstructing a simple bifurcating tree of life challenging, as genes in different parts of a genome might have different evolutionary histories. For bacteria and archaea, the tree of life might be better represented as a web or net of life. Incongruence between gene trees and species trees. Different genes or different types of data, e.g., morphological versus molecular, can sometimes yield conflicting phylogenies. This can be due to HGT, incomplete lineage sorting, where ancestral polymorphisms persist across speciation events, gene duplication and loss, or simply insufficient data. Resolving these incongruences often requires more data and sophisticated analytical methods. The pace of discovery versus description. We are discovering new species at an unprecedented rate, especially in biodiversity hotspots and microbial communities. However, the number of trained taxonomists capable of describing and classifying these species is declining. This taxonomic impediment hinders conservation efforts and our overall understanding of biodiversity. Naming conventions and stability. While binomial nomenclature is stable, the higher ranks, family, order, class, etc., are subject to revision as new phylogenetic insights emerge. This can sometimes lead to instability in classification schemes, which can be frustrating for non-specialists. Efforts are being made to develop stable classification systems that reflect phylogenetic relationships while remaining practical. The dark matter of biodiversity. A vast amount of life, especially microbial life, remains uncultured and undescribed. Environmental DNA-eDNA techniques, which involve sequencing DNA directly from environmental samples, soil, water, air, are revealing enormous hidden biodiversity, but integrating this dark matter into formal classification is an ongoing challenge. The future of classification, integrative taxonomy, and genomics. The field of systematics is dynamic and rapidly evolving. The future promises an era of integrative taxonomy, where multiple lines of evidence, morphology, anatomy, development, behavior, ecology, geographical distribution, and especially molecular data, genomics, transcriptomics, proteomics, are combined to provide the most robust and comprehensive understanding of species boundaries and evolutionary relationships. Genomics, the study of entire genomes, is particularly transformative. Whole genome sequencing allows for comparisons of vast amounts of genetic information, providing unprecedented power to resolve phylogenetic relationships, even among very closely related species. It also allows for the study of gene evolution, gene families, and the genetic basis of adaptations. DNA barcoding is another powerful tool. It involves using a short, standardized DNA sequence, a barcode, to identify species. For animals, a region of the mitochondrial COI gene is commonly used. For plants, regions of chloroplast DNA. DNA barcoding offers a rapid, reliable, and often automated way to identify species, even from fragments or degraded samples. It has applications in food safety, wildlife forensics, biodiversity surveys, and monitoring invasive species. Phylogenomics, the construction of phylogenetic trees using genome-scale data, is becoming the gold standard for reconstructing the tree of life. As sequencing costs decrease and computational power increases, we can expect increasingly detailed and robust phylogenies for all domains of life. The continuous effort to classify and understand the relationships among organisms is not merely a task of putting names to faces. It is a profound scientific endeavor that reveals the grand narrative of life on Earth, connecting every organism, from the smallest bacterium to the largest whale, through billions of years of shared ancestry. It is a testament to the elegant simplicity of evolution and the intricate beauty of the natural world. As we continue to explore, discover, and analyze, the tree of life will grow clearer, its branches will become more defined, and our understanding of the living world and our place within it will deepen. Biological classification and systematics, far from being static or archaic fields, remain at the cutting edge of biological inquiry, providing the essential framework for all other biological sciences and for humanity's ongoing quest to comprehend the richness and wonder of life.